Hi, hi. How are you doing, Kevin?
Hi, good. How are you?
Not bad, not bad. So let's give it a couple minutes for people to stream in and then we'll get started.
Cool.
If you're already here with us, welcome to our fall Research and Innovation Showcase. Hi, Jake. And
Hello,
this
how are you?
Hi, good. Thank you for being here on time.
No problem.
Actually early, but that's great. So this will be streamed over all of our Leadership Innovation Lab channels, and so this will be rerun and replayed. We have with us here our panelists and as well as our keynote speaker. I'm going to give it just 2 or 3 minutes for people to join in, and then we will get started. If you're already here, welcome. Thank you for being here on time. Lovely to have you. So, Kevin, I'm going to let you do your own screen share. Can you check that you're able to do that?
Checking now.
Excellent.
Cool.
Okay. I see that you've paused it. All right. I'm going to presume that works.
Yep.
And yep. Mhm. Okay. We're going to wait maybe 1 or 2 more minutes and then we will get started. Oh, hi, Sunny. Thank you for being here on time. Okay, we're 4 minutes past now, so I'm gonna go ahead and get started. This whole showcase is gonna be recorded and rerun. So, bearing in mind that while we may not be able to have all of our attendees join us live, this will nevertheless be broadcast across various channels. So, my name is Maya. Thank you, everyone who is here with us today. Welcome to the Research and Innovation Showcase, the fall season. And today is day one. It runs today and tomorrow at the same time. So, I'm the founder and director of the Leadership Innovation Lab, where we kind of like Y incubator for mostly high school students. I'm very happy to have you all here with us. And tomorrow is going to be hosted by one of the members of our team, Avi. And the agenda for today, we're going to have a keynote speech by Kevin. Kevin, who is a former admissions officer of Cornell University and also a cherished personal friend. Hi, Kevin. Good to have you here with us. Kevin is going to talk to us about how do admissions officers view research and entrepreneurship today, as well as some areas of research and entrepreneurship that are viewed more favorably. I had a brief glance at Kevin's content for today. I think that he's going to go above and beyond that. So he'll tell you better than I can. After Kevin speaks, we're going to hear from Jake, Angela, Christine, and Sunny about their various projects as well as research. So it's going to be a mix of research work as well as student project work today. And lastly, we will wrap up with Vanessa. So without further ado, I'm going to go over to the keynote speech by Kevin, and I'm going to let him put on his own screen share. Go for it, Kevin.
Okay, can everyone see my screen okay? Are you seeing the presenter version or
I can see it. We're good.
you're seeing presenter version or the non-presenter version?
I'm seeing the presenter version, so I see the notes as well.
Okay, so that's what you should not see. So let me fix that. The intention was to do it in the regular version. All right, how about this one? Is that better?
Just give it a second. Is it because you have to click like full screen? I don't know.
I don't know
Uh,
why it's not cooperating with me today. It shouldn't.
mm.
It was cooperating earlier.
Is it display settings? I don't know.
Let's find out. Hmm. Okay. Well, we will try it the old-fashioned way. How's
Yep, this is good. This is good.
We'll go with that. Okay. Hi,
Yep.
everybody. Thank you for bearing with me for my technical difficulties. It means I have to speak a little bit faster now. My name is Kevin, and like Maya said, I am a former admissions officer of Cornell University and a few other universities that people know of, but we'll leave it at Cornell for now. And I'm super excited to be talking with you all about entrepreneurship and research and how that is viewed in the admissions world for university. I mean, it's one thing to say that it's these are things that are important, but then to give you the other side of this conversation, I think is hopefully useful for you all, and especially as I presume many of you are either applying this year or next year or in 2 years, or maybe further out than that. It's important for you all to have kind of see how your work will be valued. First of all, I commend you all for doing all this awesome work. I took a very quick read through, and I'm super interested in some of your projects. So if I can, I'll pop in and out to hear a little bit more. But for now, thanks for having me. So for today, emissions has evolved. I think we know that there are more things than ever that are being looked at. And I can say that, especially from the Cornell perspective, but in general, how you innovate and how you showcase what you're really interested in is super key, because we're not just admitting a great student anymore. We're admitting someone who is a great student, has great ideas, who has followed through with those ideas, and then also has impact. And that ultimately stands out. There are more students than ever who are founding something, researching something, and changing. So ultimately, how will your innovation ultimately be shown? And I think your projects today will definitely come into that. There is one thing I also wish to note when it comes to innovation that a lot of students think, um, oh, when I'm applying to university, this is what needs to shine through. It needs to show that I've created something novel. Well, actually, not necessarily. We don't expect you to create something novel. What's actually expected is for you to take something that's already in place and enhance that and see where you can apply a solution to a common problem. Because then that only takes things forward. We don't have to create something brand new, but it's more of let's look at the knowledge we already have and already we know and apply it differently to create that end result. So think of it in that way as you're tailoring your projects. So I think we know that when you look at an application or an image software looks at your application, academic rigor needs to be there. But it's important to say that. Presuming that many students have that, we then look for the next bits, the intellectual curiosity, which is like everyone's favorite buzzword these days. We have initiative and, of course, authenticity. And I think that authenticity part is the one part that when we were looking at applications at Cornell, we weren't quite sure, is this research or is this project authentic work, or is it meant to look good on a university application? And that is really the difference. And so my question to you all with your research and with your projects is why? Why are we doing this? What is the reason that is motivating you behind this? If it's, it looks good on my application, that's a fair answer, and I get that. But take it the next step further. Give us that like emotion behind what you're doing to help the admissions officer understand this is why you picked this project and why you're doing it. Of course, like you probably heard many times from Maya, from Avi, and from other advisors, innovation really needs to show where your mind is at and what it is thinking. The result is one thing, but the process is very different. So I hope that you all can take a little bit from this, that it's about the process here and seeing your thoughts. Admissions officers want to get inside your brains. I know it's a scary place for people to be, especially in mine, trust me. But when you are thinking about showcasing your entire life, matter of speak, to an admissions officer, they want to know what's going on in your head. What is that why behind everything you're doing? Now, I will step back and say that innovation, this amazing word, has boomed in the admissions process, right? Many students will have picked up research, will mention this in their application, and that's great. But the pitfall to that is when it's a bit robotic and kind of expected. And before I mentioned to you one student, or not one student, but rather one particular case where it felt like this project was just kind of done for the sake of the application. Well, it's interesting enough that when you look at your project, you might be thinking, well, then who will benefit from my research? Who will ultimately be able to improve their knowledge, improve their life, improve their workplace, whatever it might be, from what I'm doing? Take that answer and try to apply your project somewhere else. I'm sure you're probably already doing that in some way, but take this next step further. Take it away from your computer screen to take it out into society, because ultimately that's when any research that's done in any way is actually going to be effective. Just keeping it on a piece of paper or on a Google Doc in your OneDrive isn't really going to help you out, right? But how are you going to apply this going forward? That's the depth behind it all. And that purpose-driven work is really key. I'll give you an example of a student I worked with a couple of years ago. She was very passionate about domestic violence, and she was very passionate about also computer science. Now, putting those two things together it's not quite easy to put those two things together. What she ended up doing was creating an app for survivors of domestic violence to better understand what they were facing in terms of symptoms, in terms of ways to get help. And she ended up creating this app that was secure enough where they were the only people who were able to access it. And then that gave them more resources, more opportunities. And then she went to a local shelter and organization to have this app be rolled out. And it was rolled out across her state, which she was in, I think, California at the time. So huge state, I think around 15 different places. And that's the purpose-driven work. That's the impact and that's the application. So ultimately, this is where it goes beyond just creating and coding an app and interest in domestic violence all the way to that purpose-driven work. So just some ideas for you all to think about as you are going forward. Now, on the other side, we have entrepreneurship. Now, I think we know that's initiative, leadership, problem solving, because everything that is created by entrepreneurship is solving a problem in some way. What I think is really key when you think about impact of this is, okay, we have this amazing idea. Is it going to be more of a social impact, a creative impact, a technical one? What might that look like for you? And I encourage you all to think about this because many students think, oh, we have to make a profit of some kind. Oh, we have to get this to be rolled out to 1,000 people. Well, I'm not so sure. When we were looking at applications at the university side and we saw new nonprofits or new organizations, we were wondering, great, this sounds awesome, but what did you do next? Who would benefit from this? And while this does not always come through naturally in an application, it does come through when you're asked about talking about it. And so even beyond, let's say if you think of an interview or you think of an essay you have to write, think about what if someone came to you and said, okay, Maya, what did your project do and who did it benefit from? If you're not really sure where that's going, then it's find that answer. If you know where that is, then how do you take that and that increase that benefit? And what type of impact is this? If it's just for the sake of knowledge, okay. But what is to come in terms of the future? How are you going to lead in the next step? Of course, Maya does amazing things, so I'm not saying she would do that, but because she was the first person I saw on my screen there, I just picked her out. So thanks, Maya. True research I think is something we often look at in university applications. And whether I'm advising students now, whether I have looked at applications before, new questions. Please pick something new, something fun, something that actually shows how curious you are. Because ultimately, it is these new, fresh questions that get people intrigued at what you're doing. Like I said before, it's not about reinventing the wheel, but it is really about showing that, okay, let's take something that we might already know and turn that example. Be persistent with it. Take it that next step further. If you think, oh, this is going too far, please go too far. Because that's when you learn about where is your comfort zone, but also where can this research be pushed. Maybe you'll find something totally new by going out of that comfort zone. And that can often outweigh duration, because research projects can't go on for 3 years when you guys are in high school, and that's okay. It doesn't have to be with, let's say, a massively large university or a professor from insert university name, which might be prestigious, to have importance. The persistence, the depth, the analysis is what's really key. Now, of course, mentorship helps, and they've had some great mentors in this program, but also owning your own work and reflecting upon that work. The best researchers can reflect on their writing and analyze what they've just done and how that is ultimately going to impact themselves as well in some way. It's hard to reflect. I think we know it's kind of hard to do that, especially now when you want sort of instant gratification. But that reflection matters the most. And that will be the part that I think is the next step after this showcase is, what feedback have you gotten? What advice have you been given? What have you learned from others? How can you then apply some of those small pieces of knowledge to increase what you've already done? That's the next step for you all, and might be even the hardest step going forward. So now you might be thinking, okay, I have some ideas here. I have some tangible outcomes from my research. What do we really want to show the admissions officer? Sustained commitment and evidence of growth. Now, I mentioned just before that duration is questionable. Well, sustained commitment usually means really devoting a certain amount of time over a period of time to what you're working on. 3 months, 6 months can be great, but ultimately, how have you grown over time? Where did you start and where did you end? And what was your thought process throughout that journey? If you think of creating an art piece, you don't create an art piece just like that. Well, I hope maybe some of you do, but I personally don't. I have to draw things out. And that sketching and that drawing of the art piece is exactly what the admissions officers want to see, because that's your growth. The end result looks great, but we can see that it looks great. But what happened before that? I've already mentioned about reflecting. I think, and this is maybe just sort of unwarranted life advice, is that we all learn lessons from everything we do, including those challenges. We have challenges at many different points in time. The level of stress will go up and down. But what was the lessons from the challenge? In so many cases, we hear about perfection and we hear about wanting it to be perfect for that university application and perfect for this showcase on October 25th. Yet sometimes the imperfection is actually what is kind of the moment of like, yeah, this is what we really can polish to make it look great. And that is often what we were excited to see at Cornell, was the imperfections, because we know that there is room for growth there, and we want to see where you all are going to take that. I've already mentioned about community impact, but tangible outcomes, whether it's from your research, whether it's where you're going to go next, continue to keep a log of that, because you can also see the growth. Maybe you're working on a project that requires social engagement, and you go from 1,000 users to 10,000 users in 3 years. That's fantastic. Showcase that. And again, like I said before, have that authenticity behind everything that you're doing. There is a difference between entrepreneurship and research. I've kind of outlined them here. But what I want to ultimately showcase is that intellectual vitality. And this part is super key, because when we are thinking about this intersection, which is very natural to have, entrepreneurship and research, you might be wondering, how can I break these two things apart? And how can I then also bring them back together to be, working hand in hand? Remember that entrepreneurship is you showing the, let's say, ideas and moving it forward and taking this on and spearheading it forward. But the research is more of the small steps to your end result. So I would look at this as like a sandwich. Entrepreneurship is kind of the sandwich, the bread on the outside, and research is kind of all the good stuff in the middle. Whatever you want to put in the middle is up to you. But what I'm saying is this is kind of how you can look at it. And so if you are thinking about how do I package this, think of your bigger beginning and end as entrepreneurship, your initiative, your ideas, how you're going to lead it, how you're going to finish it. And the research is all that inquiry in the middle. If there's not so much in the middle, that needs to be enhanced, of course. And like I said, intersection is often the most compelling. That's where all of these two things come together. Think of it like, again, I'm also hungry, right? So that's probably why I'm mentioning a sandwich. But that kind of desire to see the full sandwich and kind of completing all that is what they really want to see. They don't want to see just the bread or just the pieces kind of separate. Anyways, moving on from my hunger for now. So telling your story within the application. Tell us why. Tell us how. Tell us not just the what, but explain those struggles and show that vulnerability. Don't be afraid to say that you made mistakes. Don't be afraid to say that you didn't know where to go in your research. Don't be afraid to say you had to seek out multiple opinions, or you were stuck somewhere, or you didn't know how to get your research out to the public. That's fine to say you don't know. It's probably the scariest moment in life to say I don't know something and kind of feel a little bit lost. But it's that part that really will let you find different perspectives, showcase some of those things, show those learnings, those reflections, personal connections. And if you have the opportunity to be interviewed for a university application or be able to write about this in some way, connect the innovation, connect your, let's say, vulnerable moment to your goals, because that's where growth really ultimately can come through. And again, try your best to quantify the impact as much as you can. Now, I have 3 minutes left. So I have a case study, but I'm going to have to pass over it for the sake of time and go into the future. The future of innovation in admissions is now an academic identity. Your research, your entrepreneurship, leadership is no longer just hidden in an activities list or 10 things you have to list. It's no longer hidden in your resume somewhere. It's going to be that overarching theme that everything you do needs to hit. It's going to be kind of like a, for lack of a better phrase, like the umbrella will now be innovation. And then what do you do under that umbrella ultimately will be your pathway forward. Universities are looking for cross-disciplinary. So if you are thinking of something, let's say that is focused on AI, which I imagine some of you might be, that's fantastic. But then where are you applying that in multiple different areas? I picked out AI, sustainability, and social innovation because you can look at AI and utilize AI to then apply it socially, get people trained on how to use it, get people aware of how to use it ethically, unethically, et cetera, and then tie in the sustainable aspect. And that's going to be one part of that cross-disciplinary side of things, right? Because you have some sociology in there, some anthropology, probably some language, some cultural studies, and then, of course, technology, all tied into one. That is one very brief aspect of cross-disciplinary research and the next steps. So some takeaways: be authentic, please, with whatever you're doing. Impact does beat prestige. Curiosity and action is what we want to see. That's your innovation. Don't be afraid to push those, let's say, questions you have. Depth, reflection is paramount. It's those steps along the way in your journey, not so much the end result. And remember, those students that are admitted to the universities, whether they're the top ones or maybe not so much, are those who are building on thought, not just the application. You can apply your knowledge. Okay, that's great. But that's one-to-one. If you're building, that's like one to two to three, and that's all connected on. So hopefully this was somewhat of enough. I'm out of time, unfortunately, right on schedule for Maya. But thank you everybody for listening to me for this about 18 minutes or so. Maya, I saw a message in there about questions. Do
That's
you want?
right.
Do you have
Yeah.
some?
I saw, thank you firstly, Kevin. And I saw that there was a question from Luke. Luke, do you want to just go ahead and shout out your question, or if you want to type it out, that's good with me too. I'm gonna leave the app to you. Luke, Are you still here with us? I'm actually not here if I'm not sure if he's still here with us. That being said though, Kevin, I hate to have you skip over your case study. You want to quickly talk about it?
Oh, yeah, sure. Let me just get back to that slide because I had written it down. I want to make sure I don't get it wrong. Yes, okay. All right. So the case study I had was from the student. The student was in grade 10 at the time and did a research paper on essentially the climate of a region of the country that she lived in for a science fair. Okay, very interesting. And that research had some data that was gathered and predicted that in 10 years, because of climate change, there would be severe flooding in that area. And so she felt like this is just a science fair. Okay, whatever. No big deal. Maybe this data isn't right. But because she felt like this could have an impact in some way, maybe not when she's living there, but 10 years from now, maybe when her siblings are living there or whatnot, she went to a local engineering company and said, is this valid? And can they design a flood alert system that's at low cost, which can utilize this data to understand the flow of rain and ultimately how that could impact the area? And they use open source weather data to not only adopt this in the community, um, but then adopted by two other communities nearby. So three communities had this flood alert system put in place by the next year. And all this began from a science fair research project. And the key part was not just the scientific insight, but the action of taking it off the paper and bringing it outside when she didn't quite know how to describe all of these things. And obviously, this is a prediction as well. So you have to kind of buy into the data and have it backed up. But it was backed up. It showed initiative. It showed social impact beyond technical skill. And ultimately, this is something that was in the application that I still remember to this day. And as you know, I was at Cornell some years ago, a few. That research only mattered when someone uses it. What you said. This only mattered because the three communities actually used the research. Otherwise, it stayed on the paper. And because we're talking about research and entrepreneurship today, there was alignment there of the rigor of environmental sciences and geography to being entrepreneurial and to be looking at things beyond just what's on the resume, a transfer of learning, so to speak.
Thanks for that, Kevin. Now, I'm not sure if Luke accidentally hit the hands up button or actually had a question, but I've invited him to share his question if he does have one. For now, I'm going to say thank you very much for sharing that, Kevin, and I was very happy to be your appointed student for the purposes of this presentation. We're going to move on to the next item on our agenda. And of course, Kevin, you're welcome to stay with us. That will be a pleasure. We are looking at a presentation from Jake, who's already here with us. Like I said, Jake, thank you for showing up so early. Certainly very appreciated. Jake is going to talk to us about tariffs, food access, and community health. And I believe that Jake probably has his own materials that he's going to
Yeah.
share. Go for it,
So I should
Jake.
go ahead and share my screen, right?
Go ahead. Yeah, floor is
Okay.
yours.
Sounds good. Okay. All right. Let me know when you see it.
Yep, we're all good. I can see my own
Okay.
face. Yep, go for it.
It's just loading. Okay. All right. So before I get started into talking about my research, I just want to give some background about myself and why I decided to do this research and what I'm interested in. So I'm Jake. I'm a high school student. I'm a senior this year in Long Island, New York. And pretty much throughout my whole life, I've been very interested in just nutrition and how the different foods you eat affect your body and how it affects your health. And I think over time, I began to apply that like on a broader scale and think about, okay, how does this food affect myself, but also how does this food affect other people as well? And this interest began when I was tutoring at an after school program. I've been doing it for a few years now within my own community. And my community, I would say, is definitely a more middle to upper class community, and I was never really aware that there were, people who lived in my community that struggled with food access and struggled with access to proper nutrition. And many of the students that I tutored in the program, as it turned out, were on, federal programs and were on food stamp programs at school. So I think that inspired me to, think about how I'm applying this in my life and how I can give back to the community. And at the time, I also began developing an interest in public policy and economics as well. So my whole research is, or the basis of my research is kind of centered around connecting public health to public policy and to see how things and how different policies that have been like, as you can see, my research here is on tariffs. So how these economic policies are impacting community health. Okay, so now I'll get started on talking about my actual research. Okay, so first I'll start with the aim. So the premise of my research was to see how tariffs, which are a very hot topic right now, especially within the Trump presidency, within the new wave and implementation of tariffs, and to see how these tariffs impact food prices and also food access at gas stations. So what I did within my research is I compared low income and low access communities versus high income, high access communities across Long Island, according to the USDA's Research Atlas. So I wanted to assess the effects noted by both managers and customers, and to see with interviews at gas stations how this could impact community health. And I think I discovered that this could be an extremely or something that is very underlooked. So, but I'll get into that. Okay, so let's talk about the background and the problem. So, according to the Yale Budget Lab, the tariffs have raised the cost of imported snacks, beverages, and packaged goods. So, what does this mean, and why did I decide to do my research at gas stations? So, gas stations, and something I realized through a little bit of research before I carried out my field study, was that gas stations are a lot more important than I think people realize, especially in lower income neighborhoods, because they're the primary suppliers of food for 32 million Americans in food deserts. So, and I think through my through the interviews that I did with managers and workers at gas stations, it became very apparent to me that in a lot of low access communities, gas stations are their only source of like groceries. Gas stations are where they're getting their food from the most. And the issue with this is that when you walk into a gas station, you don't really see many like foods that you would consider to be healthy and nutritious for the body. Instead, you're seeing a lot of snacks, you're seeing a lot of packaged goods, right? So that's another issue that we see here where we have these, rising prices that are affecting gas stations. And because so many Americans in low income communities are relying on these gas stations, they're pushed to eat unhealthy foods. So that's what we see in the literature review. So Volpe et al. 2013 found that um, rising prices of healthy foods versus cheap processed ones. The cheap processed ones had a greater impact on, like due to the prices being increased, people ended up eating more cheap and processed foods, which led to worsened diets and greater levels of obesity. So to further this, the point that I made earlier, this could have a profound impact on public health as well. And the CDC in 2023 showed this, where food deserts worsen chronic illness outcomes when the prices of healthy foods rise faster than processed alternatives. So I think all this research together goes to show that like there's two key takeaways that we should take from this. The first being that gas stations are an underlooked source of food. They're much more important than people realize, especially in low access communities. And the second being that these rising prices can quickly impact health outcomes. Okay, so then that goes into my questions. So my questions are pretty much, what does this show about the inequities in food access between low access and high access um, areas, and how do the managers, workers, and customers respond to this? Okay, so in terms of what I actually did, I went to 6 gas stations, 3 low-income, low-access zones, 3 high-income, high-access zones, and I did 6 manager interviews, so 1 across each, and 11 customer surveys in total. And I collected this data over this summer in July and August, and I asked them 4 quick questions. An example of one of the questions being, have you noticed increases in the price of foods or beverages at gas stations since the implementation of tariffs? But I also asked them about like categories. I asked managers more specifically what they were noticing. And we analyzed, coping strategies, adaptation strategies to these price changes, and behavioral trends in general. Okay, so now let's take a look at the results. So the results showed us that in high income and high access stations, the tariffs were pretty much viewed as irrelevant, where only 1.17 out of 5 um, on average indicated that they depended on gas stations for food. So as we can see in the high income, high access stations, the customers didn't seem to be too affected by these results. And in the low income, low access stations, we see a higher score, but then we start to notice an issue, and the issue is the difference between the customer results and the manager results, more specifically within the low income, low access stations, where um, the managers noted that there are increases in certain categories according to the prices. However, a lot of the time, the customers didn't necessarily acknowledge that this was due to tariffs and instead blamed it on the managers themselves. But the managers noted that there were definite, well, so out of the 3 low income, low access managers, only one saw that the prices affected them immediately. However, all 3 noted that even if there weren't major price increases already, that the tariffs were going to impact them at some point. So you kind of have this disconnect between the acknowledgement of tariffs, especially in the low income, low access stations, where the customers are noticing price changes and they're attributing it to the gas stations themselves rather than the underlying cause, which is the tariffs, right? But instead, at the higher income, higher access stations, we see that the tariff effects are moderate overall. However, all managers reported that there has been less business in general. Okay, so I kind of went through I know I'm running low on time, but I kind of went through the discussion section already within the results, where the main idea is that tariffs can end up being like these hidden taxes, quote unquote, that affect people to a greater extent than many realize. And in terms of who is impacted, right, the whole point of tariffs in general is to increase, domestic markets and boost U.S. manufacturing. However, these low income consumers and these gas stations end up absorbing these price increases, and they're forced to either cut stock or raise prices and create tension, and creates a divide and impact community stability, right? So in terms of some solutions and future directions, I think in terms of short-term solutions, it's important to make sure that people have an awareness and understand what's going on around them, so increasing public education. But in terms of long-term solutions, I think it's important to look at these agreements, US, Mexico, Canada agreement, and to ensure that we don't want to have like this double-edged sword where, yes, tariffs could be doing something to boost US manufacturing, but at the same time, they're also impacting low access communities, right? So I kind of touched on these research implications already. So let me go ahead and talk about the limitations. So obviously we have a small sample size and it's located in only Long Island. So we don't really have like a generalizable idea, right? And obviously tariffs are a hot topic right now. So there's always going to be changes. And I think the biggest issue is that we don't have distributor level pricing data available, right? So we don't know exactly what the price differences are. We're just going off of word of mouth and what people believe. And I think we also need to have more behavioral data in the mix as well. Okay, so to conclude everything, gas stations are often the primary source of food for many low income, low access neighborhoods, and this has become something that's been extremely overlooked. So there could be health inequities related to this as well, because foods at gas stations are not necessarily the healthiest. And there was this clear disconnect between the managers and customers, especially in low access communities where there were price changes observed. However, the managers knew that these were due to tariffs, but the customers didn't. And this ends up creating a divide within the community, right? So overall, policies must be made to balance this idea of economic protection and boost U.S. manufacturing, but they should also be maintain like they shouldn't be taking away from vulnerable communities, and they should be there to also protect basic human rights such as nutrition security and affordability. Um, and then these are my references. And yeah, thanks everybody for listening. That's it.
Thank you very much, Jake. That was great. I, of course, invite you to stay for the other presentations, and thank you so much. Let's move on to the next one, which I believe is Angela. Angela, I see that you're here with us, and you're going to be talking about navigating narratives. I'm very happy to see you talk about this project, Angela, given the amount of time that we've spent growing this project together, and I was very happy to accompany you on that journey. So I'm going to give you the floor, Angela, and you can go ahead.
Thank you, Maya.
You're welcome, dear.
Is it?
Yep, we can see it and I can hear you clearly. Go ahead.
So navigating narratives is a project that is relatively new. And because I'm pretty interested in visual art and storytelling, I really want to combine those two. So so this phase mainly focused on the Dupont grottos, which is in China. And so there are like a lot of different like Buddhist murals and sculptures. And I really want to focus on like the storytelling aspect and maybe also like the historical aspect of those artworks. And my aim for this project is just to share it with a wider audience so people gain more knowledge on the subject. So this project just kind of like I said before, combines research, creativity, and storytelling. So I really want to lean more into like the human aspects of it and how it is like important for people to connect to like their cultural history and which I will also talk about later in the presentation about a previous exhibition I did, also relating to like cultural history and oral history. So one of the challenges I faced in this project is that I have like a variety of interests and they change a lot over time. So I really wanted to try and connect all of these together so that it's like more unified. And also for the previous exhibition, I found it quite challenging to like communicate with all of like the students so that I can collect their artworks and make sure that the process is smooth. So like I said before, I had a exhibition in 2025 in my school where I think around 18 artworks were exhibited in my school, which was based off of the theme Outsiders, which was also like the English course theme in my school last year. I mainly gathered artworks from students outside of my school so that they could focus on like a variety of perspectives. Um, it also connected to an organization I had previously, and I still have it, but it's on pause right now. Where we kind of interview people. Um we did several interviews in West Queen West and also Chinatown. So we kind of summarized our findings and portrayed it in a visual way. So that was presented in the exhibition. And I am hoping to curate another exhibition, this time both in my school and online, featuring Dupont artworks this time, and focusing also on like the storytelling and like the significance of each piece. And also to highlight like I think that like Dupont, like there's a lot of different styles, and because it's ranging from a long period of time, I think that it's really worthy to look into that. Um, and because this is relatively new, I'm still working on this exhibition, and I'm hoping to also have host a symposium with professors and students talking about like art history and maybe also like focus on Dupont art. And also in the future, um I may publish a research paper also on Dupont art, and like I'm hoping to narrow down the subject a bit more. So thank you for listening.
All right. Thank you, Angela. I am aware that it's a project in progress, but that's fine. I think that you've come very far in a short amount of time, and the project has a lot of growth potential. So I want to thank you for sharing what you've done so far with us, and I look forward to us continuing to build the project together. I'm sure that you and I will talk about this very soon. I'm going to move on then to Christine. Christine, who's going to talk to us about major depressive disorder. Now, Christine, I'm sure that you have more details about your topic, but all I got was a MDD, but I'll let you talk more about it. So go for it, Christine.
I'm like not home right now, so it might be noisy later on. So I'm sorry about
No,
that.
I hear you crisp and clear. Go for it.
All right, so just a little bit about myself before I get into like the complicated stuff, I guess. I'm currently a senior in high school. I go to Hunter College High School, and a lot of what my research is inspired by is sort of my classmates who do have depression, and I've like worked through sort of like their concerns about their own medication and stuff. So a lot of this is finding a new way of targeting depression and sort of this thing that people have looked into, but sort of haven't expanded a lot yet on as of like my research. So a lot of this is a really long title, and I'll get into like the complicated stuff and I'll explain it. So again, my research aim is to investigate something called histone deacetylase 5, which is HDAC 5 in major depressive disorder, and how sort of epigenetic mechanisms can be leveraged for clinical translation. So, epigenetic mechanism is just like sort of how our genes or how our environment can affect our genes that eventually affect sort of depression and depressive outcomes. So this is like a table of contents, but I think I'm gonna like shorten some of it just because of time. So I'm just gonna go over what major depressive disorder is and like the impact of it. So there's over 163 million people in the world affected by major depressive disorder, and according to there are 322 million people with depression globally. So current therapies are standard drugs like SSRIs, SNRIs, and tricyclics. So basically what you need to know from this slide is that about 40% of major depressive disorder patients given these drugs are either unresponsive or have other side effects. So this might be like delayed onset or just more chances of recurring depression. So this is when we get to epigenetics. So epigenetics means like gene regulation without altering our DNA sequence. So a lot of this is stress and trauma leads to sort of histone modifications. So examples of which are methylation and acetylation. So that's like how tightly DNA is wounded around histones. And I guess like for those who haven't taken like biology, it's basically like why your eyes don't make like insulin, let's say. It's because your genes in your eyes are so tightly wounded around histones for that gene to make insulin that your eyes don't like do that. So basically more acetylation equals looser DNA and more active genes. And HDACs remove acetyl groups, causing tighter DNA and suppression of gene expressions. So that's essentially that slide. So this is on like histone acetylation and depression. Basically, chronic stress sort of leads to more acetylation, which means there's less something called BDNF expression, which just basically means like you'll be more stressed, you'll kind of have you'll display more like depressive behaviors. So I'm just gonna go over like the classes real quick. The one we're focusing on is class 2. It's on HDAC 5, mainly because HDAC 5 is regulating this sort of like stress model of depression rather than some of the other HDACs. Yeah, so these are sort of like preclinical drugs. Okay. So some of the challenges with HDAC inhibitors and its use in clinical usage is 3 main things. One is toxicity. So some HDACs affect non-histone proteins. So there's like P53, which is related to cancer. So it's sort of issues on toxicity. And then the second one is sort of its effectiveness, which is limited by the blood-brain barrier penetration. So there are certain drugs such as SAHA or valproic acid that like, yes, work, but it can't get into the brain such that it's very effective. And even if it gets into the brain, we need higher doses just to have that chance of it going into the brain, which makes it more toxic. So it goes back to that toxicity issue. And then there's also side effects that we don't know really yet, which affects sort of GI toxicity or cardiac events. So overall, there's sort of a need for more selective HDACs and better deliveries, which I will explore later on as well. So I guess I can skip on those. So some targets include BDNF, CFOS, and NPAS4, and these impair neurocircuits and basically reduce reward signaling, which leads to depression behavior symptoms, essentially. And this is just like essentially the mechanism. So HDAC5 literally acts as a bridge between stress and depression. So repressing vital genes reduces neuroplasticity and triggers more depressive behaviors. So decoding these pathways opens sort of new therapeutic doors, and we can sort of have these more personalized treatments for people with depression. That's basically what it says. So current research that I've looked into have evaluating animal models. So this is going to be really quick just because it's not like the main thing. So animal models sort of, they use these different tests to look at the coping and the reward signaling for rodents. So each has sensitivity and reproducibility issues. So again, the limitations in screenings are that they're not full models and can give more false positives because of sort of depression being very different in rodent models versus human models. So this is just like mouse models essentially. And so we sort of look at that like rodents can't model subjective symptoms like suicidality or complex cognition as human depression often does. And also most animal studies are male only. So they're sort of restriction or limitations in that sort of study as well. Okay, I think I can skip over this. So this is on like more clinical trials for like humans as something. So the main thing with, I think, depression trials, so the main challenge is really the placebo effect, where up to 40% of patients show improvement without being given sort of the drug. So what that kind of, I guess, signifies is that like it's not the treatment that's working, it's sort of not really like, it's kind of like the mindset, but sort of like how stressed they are, but also more environmental impacts on the patient's health rather than the drug being something that's like helping. So I can skip that. Sorry. Okay. So I'm gonna go on to the blood-brain barrier, which is sort of the main, I guess, pitfalls of our delivery now of HDC. So this is again like why we should target depression through epigenetics. And so the main thing is SAHA that boosts excitatory transmissions in animals, but fails clinically in humans due to poor blood-brain barrier access. So basically your brain it has a bunch of these tight junctions and efflux pumps, and sort of it restricts drug entry because you don't want like foreign substances going to your brain and like messing you up, I guess. So most HDAC inhibitors show low plasma to brain ratios, so solutions have to be addressed about the blood-brain barrier. And then SAHA's polar structure blocks brain entry, while valproic acid, despite good penetration, comes with broad toxic effects. So high doses are needed for central nervous system impacts, which often, again, raise that toxicity issue that we were talking about. So some strategies that were looked into were something called focused ultrasound, as well as prodrugs, nanoparticles, and blood-brain barrier modulators are also promising as ways to boost drug delivery to the brain, making therapies more safe and more effective. Another thing that was looked into is something called PET imaging, and it sort of reveals which HDAC inhibitor can actually reach the brain. So most show sort of poor intake and require structural tweaks, but PET imaging is incredible in sort of guiding novel drug designs for central nervous system disorders like major depressive disorder. So some future directions that we must go into is sort of developing blood-brain barrier permeability, as well as isoform-specific HDAC5 inhibitors, and also test new methods for non-invasive blood-brain barrier access. So we're going to isoform-specific targeting. So again, all of this is to show like how we can target one specific HDAC without affecting everything else in your brain that might be normal. So I guess what are isoforms? So isoforms are variants of a protein with unique function and locations. So targeting specific isoforms can have profound therapeutic benefits while sparing others. So PAN inhibitors cause toxicity, and isoform selective drugs means precision and safety. So selectivity is already proving effective in cancer drugs and holds promise for depression. And that's like the overall again. And sorry. And basically, like overall, selective HDAC5 inhibition is promising in research right now, but typical HDAC drugs don't enter the brain well. So again, innovative strategies like LAT1 prodrugs, nanoparticles, or other forms of delivery should be under development and should also be looked into. So some next steps are again, just overall, developing brain penetrant isoform selective compounds, as well as finding robust biomarkers and translating mouse findings into human trials. So this overall will help to sort of make HDAC a important role in depression and finding personalized treatments so that those treatments are more effective in people with depression. And so that's basically it. So thank you. And thank you to Dr. Ted Sawyer, who is my professor, as well as Ms. Maya G, who allowed me to present my research.
Oh,
Yeah.
aren't you sweet, Christine?
Sources.
Thank you so much for talking about that. No, I was never really a STEM student myself, so I think a lot of what you talked about already like went over my head. I did follow it, but I'm honestly more of political science myself. Still, I think what you did was very, very impressive, especially at your age, and I want you to know that, honestly, well done. Thank
Thank
you
you.
for, no, you're welcome, of course. Thank you for presenting here with us today. We have one more research to get through from Sunny about cart therapy, and then I'm going to be talking a little bit about Vanessa's business fashion business project. Sunny, hi. Floor is yours. Go for it.
Okay, sorry, give me 1 second to present this. Sorry, can we all see this?
Yep, all good.
Okay, perfect. So to start out, I guess I'll introduce myself first. My name is Sunny Ma. I'm currently a sophomore in high school, and I'm interested in biology, neuroscience. I'm interested a lot in the brain, and I'm also interested in different cancers. Growing up, I've been exposed to this kind of work a lot. I have family that does research in both cancer and neuroscience, so that's kind of where my interest developed from. Today, I will be presenting to you guys about CAR T cell therapy in treatment of neuroblastoma. So first, I would like to explain what exactly is neuroblastoma. So this is the most widespread extracranial pediatric solid tumor, and it's usually found in children that are under 5 years old. And it actually makes up 15% of childhood cancer deaths, even though neuroblastoma or NB only makes up 8 to 10% of these cancerous cases in general. And this is a disease that comes from our parasympathetic nervous system. It's generally found in our adrenal glands, and it forms due to failed differentiation of our neurocrest cells. So basically, when these embryonic cells fail to complete their proper differentiation, they can create these clumps in our body and just clump together and form these solid tumors that can grow there and become really fatal and damaging to our body in the future. And there are a lot of different reasons why neuroblastoma is so severe and so hard to treat. The first is that it displays high clinical heterogeneity, which basically means that NB tumors come in many different diverse shapes and forms. So as you can imagine, that makes it hard to develop treatments for it because there are so many different kinds of it. Additionally, NB also displays high rates or frequent metastasis, which basically just means that these tumors, once they develop, they are constantly and continuously growing, developing, expanding, and moving around, expanding to different parts of your body, which again can make it really hard to track down and completely remove it from your body. Additionally, NB cells also have really low expression levels of different molecules that help your immune system detect these tumor cells. So, for example, MHC-I protein and different neoantigens are both molecules usually expressed on the surface of these cells that help immune cells detect, cells that we want to eradicate from our body. However, NB cells have really low expression rates of such molecules, which is why it has such a low immunogenicity level. It's really hard to detect them, really hard to kill them afterwards as well. And so a couple past methods that have been used to treat NB are surgical removal, chemotherapy, and radiotherapy, and a couple others. But a lot of these methods don't actually show super great or super effective results. Oftentimes, high relapse is still seen in patients. And so today I'd like to focus on a different type of immunotherapy, CAR T cell therapy, which is a more novel method that is, still expanding. It's still a growing field, but it's definitely showed a lot of promise for the treatment of NB. So just an introduction on what exactly CAR T cell therapy is. This is basically a form of solid tumor immunotherapy that takes the T cells, the immune cells from the afflicted patient's bodies, and they are engineered to express these molecules called CARs, which stands for chimeric antigen receptors. And what these things do on these CAR T cells is that these CARs are able to then recognize antigens found on the surface of tumor cells. And when they recognize these cells, these CARs help the CAR T cells, the engineered T cells, identify, then latch onto the cancerous cells. And once they are attached, they can be used to kill the cells. And in this case, scientists use them to kill these neuroblastoma cells. However, there are definitely some challenges that come with this method, even though it shows great promise. The first is that there is a really diverse expression of different tumor-associated antigens, TAAs for short, that is found on NB cells or other cancer cells. And that makes it really hard to identify TAAs that are specific to NB, because if we want to most effectively treat this cancer, the best case scenario would be to find a TAA that is explicitly found on NB, so we can just hone in on it. However, that's really hard to do. Additionally, tumor cells, NB cells, like all tumors, have immunosuppressive tumor microenvironments that surround the tumor cells, and the job of this environment is to basically protect the tumor, to keep it alive, and it does this by hindering and obstructing any sort of immune cells that try to make its way to the tumor and attack it to kill it. And it's really hard for T cells to survive in this environment, and it's really hard for T cells to traffic to the tumor due to it. And oftentimes it is seen that CAR T cells can lack the endurance or persistence that they need in order to survive and traverse this environment. And they can often get prematurely exhausted, and so their effects are, not as good as we want them to be. Additionally, there are two main adverse side effects that can be seen as a result of CAR T cell therapy. The first is cytokine release syndrome, or CRS, which is basically an excessive release of inflammatory cytokines, which are signal proteins that your immune cells of your immune system, will release into your body, and they're super important for coordinating your cellular functions within the immune system. But in this syndrome, an excessive amount of them are released, and not only that, but they're inflammatory. So they can cause inflammatory damage to your tissues, your organs. A lot of side effects or symptoms can be felt, such as fever, chills, headache, etc. So this can definitely cause long-term damage to your body, and is something that, is actively being addressed. Additionally, on-target off-tumor toxicity is another adverse effect, and this is basically something that happens when healthy tissues are damaged by CAR T cells. And this is because it is incredibly rare for solid tumors to have tumor-specific antigens. Usually antigens that are found on tumor cells can also be found on healthy tissue. So as you can imagine, this can lead to CAR T cells not just, targeting our tumor cells, but our healthy tissue as well. So that's another thing that different scientists are definitely considering in their research. So we're going to be spending a couple of time on this slide, where I've kind of just briefly listed the different treatments that are currently being prominently researched, but I'd like to go into detail on each of these for you. So the main approaches include um, targeting different antigens. And the first one that we're going to discuss is GD2. So GD2 is a ganglioside. I know it's a big word, but basically it's a form of lipid that is found on the surface of these tumor cells. And what's important about it is that it functions in helping with tumor immune evasion and suppressing immune responses. So that's what makes it a good target for us when we're trying to enhance our immune system and kill these tumor cells. But a setback to GD2 is that it's not specific to neuroblastoma cells. It is found on healthy tissue as well. And as a result, scientists have found that research or methods targeting just the GD2 ganglioside on cells often has low remission and also high relapse in patients. So it's definitely a promising method, but of course has its own setbacks. So another antigen that scientists are looking at is GPC2, which is just another antigen, another molecule found on the surface of tumor cells that functions in enhancing tumor growth, which is what we don't want, which makes this another good target to latch onto, to target, destroy, so that we can therefore destroy the tumor cell. And what's specific or unique about GPC2 is that although it does have relatively low expression levels compared to maybe other antigens, it is specific to neuroblastoma cells. So that definitely makes it easier to hone in on the tumor cells that we are trying to target. Finally, CD276 or B7H3 is another prominent target that is being considered. This is an immunoglobulin molecule that is important to our immune system. It basically just boosts the function of our immune system. And what's unique about this antigen is that it shows limited expression on healthy tissues. So as you can imagine, this reduces the possibility of on-target off-tumor toxicity. So that's definitely a really good benefit of it, something that we want. And they have been found to be really effective when CAR T cells have been designed to target CD276. So those are some prominent antigens that are being researched currently. Another method of enhancing CAR T cell therapy is developing CAR T cells using natural killer T cells, which is basically just a specific line of T cells, natural killers. And what's unique about them is that they possess receptors for chemokines, a specific type of cytokine that are secreted specifically by neuroblastoma cells. And these receptors are something that regular T cells do not possess, which is why natural killer T cells can provide an additional benefit. And they are capable of efficient trafficking to tumor and metastatic sites. And they're also able to kill macrophages that propagate tumor growth. So they've been found to be able to productively regress tumor growth. So that's definitely a good avenue that scientists are exploring. Additionally, another thing being researched is IL-15, which is a cytokine that basically just contributes to the function of different immune cells. And scientists have found that when these CAR NKTs, CARs designed with natural killer T cells, are modified to secrete IL-15 at the same time, the effects of these CAR T cells are basically amplified, and we can show better resistance to tumors. So that's just, a pathway for improving upon our existing methods. Next, I'd like to move on to autophagy, which is basically a self-degradative process that cells have. It's a form of recycling. It keeps our cells clean, and most importantly, at least for this topic, autophagy also assists in tumor survival, which is why scientists have explored the inhibition of autophagy to reduce tumor survival. And this has been fairly successful, as scientists have found that by inhibiting autophagy, they have seen reduced growth of neuroblastoma spheroids, and they've also seen increased expression of different proteins on tumor cells that make it easier for them to be identified and killed by our immune cells. And so, all in all, this has made our neuroblastoma cells much more sensitive to CAR T cells, which has, of course, made CAR T cells more effective. And similarly, inhibition of YAP, the yes-associated protein, which is our final method that we'll be discussing today, has also shown good results. The yes-associated protein is a co-regulator of transcription, so a key step in how we express the genes in our body. It's a co-regulator of that in our body. And it is expressed very highly in neuroblastoma cells, which makes it a good target again for our immune cells, and it contributes greatly to immune resistance within the tumor microenvironment. And specifically, it suppresses the expression of GD2, which is what we don't want. We want high levels of GD2 expression so that we can better and more easily target such antigens to kill the cells. Therefore, scientists have experimented with inhibiting the yes-associated protein. And with this, we have found that the effects of immunotherapy targeting just GD2 have been enhanced. So we've seen better results of CAR T cell therapy by inhibiting YAP. So that was a lot of dense information condensed into one slide, but that's a general overview of the most prominent approaches. Next, I'd like to just briefly discuss some future perspectives, pathways that we should consider when improving on CAR T cell therapy and NB treatment. The main concerns, of course, is that we need to ensure the safety of patients and reduce the likelihood of the adverse effects while still trying to keep our experiments accurate, effective, efficient at removing tumor cells. And this is really hard to do as we touched upon earlier, CAR T cells, there's a lot of different challenges and obstructions in the way of making it both effective and safe at the same time. This has led to the development of different means of regulating CAR T cell therapy and forms of immunotherapy. Different scientists have developed, for example, things like synthetic regulatory circuits, which are basically mechanisms that you place inside the engineered CAR T cells, and that regulates the proliferation of CAR T cells. It regulates the cytokines and different molecules that they release, stuff like that. It's just kind of a checkpoint, a means of scientists to kind of keep an eye on the CAR T cells and their potential effects. And then finally, another perspective is just that a lot of research in the past has used mouse strains, mouse tissue, because obviously that's accessible, easy to work with. It's generally considered ethical. But mouse and human tissue obviously does have structural differences. Like, for example, human immune cells actually don't traffic that well in mouse tissue, and yet we use mouse tissue for a lot of our experiments. So the accuracy of these experiments and their results is definitely something that should be considered, should be contested, and should definitely be improved on in the future. So just to wrap up kind of a very dense and lengthy slideshow of information, some conclusions that I drew in this review of NB treatment, CAR T cell therapy. There are definitely a lot of diverse methods in the field right now that are being developed, being researched, and they do show a lot of great promise. And just to kind of summarize, these methods include targeting different types of antigens, engineering using different cell lines, different types of immune cells, inhibiting different cellular processes, and just focusing on different components of the immunotherapy in general in order to, improve and enhance the effect of CAR T cell therapy. And they've definitely showed a lot of great progress so far. These are all novel methods, of course, and they're under research, but they have showed good results, and there's definitely a lot of potential for them. But future investigation, of course, as always, is always necessary to expand upon these techniques and at the end of the day, ensure the continuous safety of patients. So these are these are my references. Thank you so much for listening to my presentation. I really appreciate it.
Thank you so much, Sunny. That was great. All right, thanks everybody for your patience. I'm just going to very quickly show you something that Vanessa did. Now, Vanessa is based in Sydney, so she can't be here with us live right now because I think it's the middle of the night for her. So I'm going to be showcasing for her just a little bit of what she did. So I wanted everyone to see the work here from Vanessa. Vanessa is actually a student very interested in the marketing and business of fashion. So she actually wrote this little book about the business insider on runway fashion. So she went over the design process. I'm going to try and make this bigger for all of you. She went over the design process. She went over the manufacturing, the supply chain management, the marketing, the cost of a runway show, for example. And I believe, monetization of fashion. So I thought that this was a very impressive piece of work, especially above and beyond everything else that most high schoolers today have to do, including all of our presenters here today who did their research and their project works above and beyond everything else that they already have to cope with. So big kudos to all of you. And this is a little book that Amanda will be self-publishing on Amazon very soon. Now, obviously, the I guess the profit-seeking side of this book is not the point. I think Vanessa just really wants to be able to share her passion about the business workings of the fashion world with everyone. So this is a very good representation of how we might be able to translate that interest into a real project that could be of significant use to profile building. So obviously Vanessa's gonna go for a I think she's gonna go for a business commerce marketing major, as well as something like a minor in fashion or graphic design or something like that. And so this would fit really nicely with that profile. As a final demonstration to you all of the extent to which a signature project can really help you heighten and elevate the application persona that you're going for. Wonderful. With that, I want to thank everyone for being here with us on a Saturday. Thank you to all of our presenters, Sunny, Christine, Angela, Jake, as well as our keynote speaker, Kevin. And this is day 1 of our fall showcase, and there will be a day 2 tomorrow where we will be going through more research projects as well as more entrepreneurship projects. And we will be talking about several demonstrations from the Next Gen United Nations Leader Bootcamp from this summer, as well as going over what Lil Digital Lab can do for you as one of the services offered in our ecosystem. And finally, very excitingly, we will be announcing the winners of the Ideation Global Pitch Competition, as well as the amounts of funding that they have won. So thank you for being here with us today, and I hope to see you back again at the same time tomorrow for day 2 of the showcase. Have a lovely Saturday, everybody. Thank you.
Thank you so much, Maya.
No worries. Goodbye.